Hi, happy to see every one of you. In this video lecture, we will concentrate on the phylum Porifera. Hope you may know about sponges, which is a member of Porifera. Here there are different type of sponges, ascon type, leucon type, like that. We will see how the water current or flow of water occurs in each type of sponges, and then what are all the classes of Porifera, and at the end of the session we will discuss the few number of questions which are already asked in the AIPMT exam or NEET exam. I hope you all may be happy to see this video lecture. After finishing the session, if you like this video very much, then don't forget to subscribe it. I have plans to give more videos regarding the NEET examination, by which everyone can pass the NEET without reading any notes or materials. If you listen, that's it enough. Now we will see about the general characters of NEET. Here they are the primitive and multicellular animals having cellular grade of organization. Here I want to discuss about cellular grade of organization. Porifera is a multicellular animal but exhibiting cellular grade of organization. There are three types of grade of organization over here. One is cellular, next one is tissue, and the third one is organ system. This Porifera is made up of many cells because they are multicellular, but they exhibit the various functions: digestion, respiration, excretion, by using in the individual cell itself. They doesn't form the tissues in order to perform the particular function. So this is exhibiting the cellular grade but multicellular animal. This is very important concept in the Porifera. They have already asked in the NEET examination also. And the next thing is, very important thing is, water canal system or water transport system is present in sponges. How this system works? Here the sponges have many pores in their body. Here you can listen. Here these dots are many pores. This is cycon, and this one is euspongia. This is also having many pores. So this is the structure of sponges. If you look into it, there are three important regions. Water can enter through the ostia. Here this is ostia, and by these ostia they can enter into the spongocoel, which is a body cavity, and then this water will move outside through the osculum. This is the three important region with respect to the movement of water. Here this pathway of water is essential for digestion, respiration, and also excretion. By here the path of the water will helpful for food gathering. By means of food gathering, it will enter into the digestion, and the digestion will result in excretion. This all happens in a single cell itself. That is why it is exhibiting cellular grade of organization. And then choanocytes. These are all the cells which are lining the spongocoel. Here you can see. This is the elaborated structure of the particular region, wherein you can see the choanocyte cell very easily. What is the role of choanocyte? They can able to filter the food particles when the water enter through the ostia pores. Food particles can be captured by means of choanocyte. This is the very important role. And then the sponge's body is supported by a skeleton. that is made up of spicules or spongin fibers. Here this is spicules. Here you can see sharp body, that is spicules. These spicules is made up of silicon. Some of them is made up of calcium. That depends upon the individual. So these are all the general characters, and this is the information what we have seen about the sponges. These are all the type of sponges. This type is ascan, which we saw already, and this type is called sycon, SYCON, and this type is called leocon. So ASL, ascan, sycon and leucon. You have to remember like this: A, S, L. A for ascan, S for sycon, L for leucon. Okay, next slide. Here, this is ascan type, where we want to describe about the flow of water current. It is nothing but flow of water. Here this is the type of ascan type of sponges, where you can see many pores on the side. Here the water from the exterior move through the osteo, and then this water can reach the spongocoel over here. This is the body cavity spongocoel. By means of osculum, this is osculum. Here you can see this is osculum. By means of this osculum region, the water can move out. This is the flow of water current in ascan type. Okay. First the water can enter through the pores, osteopores, and then they can reach the spongocoel. After that the water can move outside or exterior through the osculum. This is the ascan type. Now we will see about the flow of current, water current in the sycon type. Here this is the sycon type. The water will reach the ostium. Here you can see the ostial pores are there. Here ostial pores are there. Here you can see, and then this water will reach the incurrent canal. Here this is the between two things is this is incurrent canal. Here this is incurrent canal, and then through this incurrent canal the water will move through the prosophile and reach this is prosophile. Here you can see this is prosophile. By means of this prosophile, the water will move from incurrent canal to the radial canal. So prosophile is the intermediate between these two regions. One is incurrent canal and the another region is termed to be radial canal. Here through the apophile, this is the apophile region. This is apophile region. Through the apophile, the water will reach the spongocoel. The center part body cavity is called spongocoel. Through the apophile, the water will reach the spongocoel. Okay, and then the water can move outside by means of this osculum region. This is how the flow of water current takes place in sycon type. You just have a recap. The water from exterior moves through the ostium. Here this ostium, enter into the incurrent canal. This is incurrent canal. Through the prosophile region, it will enter into the radial canal, and through the apophile region the water will move towards the body cavity, that is termed to be spongocoel. From the spongocoel, the water will move outside by means of a osculum over here. This is how the water current will flow in cyclone type. Let's go for the next type, Leocon type. Here the water from outside or exterior moves through the dermal ostia, that is the opening over here, ostia pores, into the incurrent canal, and through the prosophile, the water can reach the flagellated chamber over here. This flagellated chamber is the replace of radial canal from Cyclone to Leocon type. In case of Cyclone type, you can see the radial canal instead of flagellated chamber, where here you can see flagellated chamber. This is the incurrent canal, this is the dermal opening ostia, incurrent canal, through this it reaches the flagellated chamber. Whereas in Cyclone type, the water will reach the radial canal. Here by means of this apophile, this is the apophile region, the water will move through the excurrent canal and reach this region, that is excurrent canal. From the excurrent canal, the water will move outside through the osculum. So the difference lies over here between Cyclone and Leocon is the movement of water from ostia incurrent canal to the radial canal in case of Cyclone, whereas here flagellated chamber instead of radial canal. And also from the flagellated chamber, the water will reach the excurrent canal by means of apophile. Here two things are very important. Before prosophile, prosodus will be there. Before apophile, apodus will be there. That is also helping the apophile and prosophile to do the process. This Leocon type canal system is seen in spongilla. Now we will see about other characters of sponges. Here the digestion is within the cell, that is extracellular. That is why I am saying that it is the cellular grade of organization. Digestion is within the cell. And nevertheless, the sexes are not separate. Here see, both male and female, that is sperms and eggs, are produced by same individual, hermaphrodite. The sponges reproduce asexually by fragmentation, that is one portion of the body will cut down and the new organism will generated from the cut down portion, that is termed to be fragmentation or budding. Asexually they can reproduce by fragmentation, and sexually by means of formation of gametes, sperms, eggs also. How the fertilization is taking place over here? This fertilization is internal. Within the body, the fertilization is taking place over here. Now we will see the various classes of Porifera: Calcareae, Hexactinellida, Demospongiae. These three are three different classes based on the type of spicules they have classified over here. Calcareae spicules are made up of, that is spongin fibers, or body skeleton is made up of calcium carbonate. This is very important one. Calcareae, calcium, C itself you can indicate calcium carbonate. Hexactinida, that is made up of siliceous spicule, silicon is there. Demospongia also silicon spicules. So hexactinida and demospongia silicon spicules are made up of silicon, whereas calcareae is made up of calcium carbonate. This is very important thing. And next thing is the canal system, asconoid, syconoid and leuconoid type. All the three types are present in these classes. Here this is syconoid and leuconoid type of sponges are seen in Hexactinida. Here the canal system is leuconoid only, demospongial leuconoid only. Here calcareae is needle shaped and 3 to 4 rayed spicules are present. Here the 3D 6 rayed spicules are present in the Hexactinida, and demospongiae six-rayed spicules are present over here. What is essential? This is all are marine. Here marine, most deep water species also. This is water, most sponges belonging to this class. Demospongiae is the very important class where many sponges are belong to this class. Demospongiae, very prevalent one. And these are needle shaped, and this one is cylindrical or funnel shaped, and the third one is freshwater and mostly marine, family freshwater and mostly marine. Here what is the thing you have to keep in your mind? There are three classes of Porifera: calcareae, hexactinida and demospongiae. Demospongiae is the prevalent one. Most sponges belong to demospongiae. And these spicules are made up of calcium carbonate. These spicules are made up of silicon. These spicules are made up of silicon or spongin or both. Here this is needle shaped and this one is cylindrical funnel shaped. This one is there is no shapes, but most of the sponges belong to this class. This is the very important criteria over here. Next thing, you just know about the examples. Sycon, this is from Scypha. Euspongiae officinalis, bath sponges. Spongilla is termed to be freshwater sponges. Spongilla. So Sycon, Euspongiae, bath sponges, and Spongilla is freshwater sponges. These three things you have to keep in your mind as a member of Porifera: Sycon, Euspongiae and Spongilla. Let's have a set of questions that can be asked in the that was already asked in the NEET question. The primitive type of canal system in Porifera is primitive type or simple type is Ascon. Most complex type Leucon type. Okay. Next one, what is true about all sponges without exception? This was asked in the NEET 1966. They are marine. Option B, they have flagellated collar cells. Yes. And the third one is mixed skeleton made up of spicules and spongin fibers. They have reproduced only asexually by budding. No, we have deal with sponges. No, all sponges are marine, some are freshwater. So first option is not the answer. And the third option, they have mixed skeleton made up of spicules and spongin fibers. This is in the case of demospongiae, but not in Hexactinida and also the class calcareae, which is made up of spicules only. So that is also wrong. They reproduce only asexually by budding. No, they reproduce by budding asexually, and in the meantime they can able to reproduce by means of sexual also. Okay, which is right? They have flagellated collar cells. Collar cells means choanocyte. All the sponges must have collar cells. Here you can see the collar cells choanocyte. Here also you can see the choanocytes. Here also you can see the choanocytes lining the spongocoel. So what I want to tell, every sponges must have the flagellated collar cells or choanocytes. Cycon, third question, cycon belongs to a group of animals. Cycon, yes, at last we have seen the members cycon, and then Euspongiae, and then Spongilla, freshwater algae, freshwater porifera sponges. So cycon belongs to a group of animals which are best described as, this was asked in the 2003 question. Multicellular, we having tissue grade. No, there is no tissue grade here. Unicellular or acellular. No, this is multicellular. This is multicellular without any tissue organization. Yes, this is right. Here, even though it is multicellular, there is no tissue organization. Cellular grade of organization is over here. Third option is right. Here, second option. Here, first option. And the next set of question, classification of Porifera is based on what? The very important question I already discussed. The classification of Porifera is mainly based on spicules, not on symmetricity, not on reproduction, not on branching. It is mainly based on spicules. If the class is Calcara, then the spicule is made up of calcium carbonate. In case of others, Demospongia or Hexactinodon, the spicules are made up of silicon. If you think this video is right and opt for NEET preparation, then don't forget to subscribe this video, because we have plans to publish many number of videos, especially focusing on the NEET questions which are already asked in the previous examination. So you can score good marks by just visualizing this video. So don't forget to subscribe this video and be a part of us and enrich your knowledge and get success in the NEET examination. Thank you.